Once each collection syringe is filled with lipoaspirate, replace the harvester cannula with a tip cap on the end of the syringe. With the tip cap facing down, place the syringe vertically into the decant rack. Allow the lipoaspirate samples to stand for at least 1 minute. The infranatant fluid will settle to the bottom of the syringe. Expel the separated infranatant fluid out of each sample syringe into a disposable cup. Prior to transferring lipoaspirate to the process disposable, be sure that the disposable plunger is completely depressed and flush with the lipid barrier disc at the bottom of the syringe. Attach a female-to-female connector to the collection syringe. Attach the process disposable to the collection syringe via the female-to-female connector. Position the connected syringes so the collection syringe is on the bottom, vertical to the process disposable. Expel the lipoaspirate sample into the process disposable. Repeat the sample transfer steps for the second collection syringe until the process disposable is completely filled. If the process disposable is still not completely filled after transfer of all the lipoaspirate, fill the remaining volume with sterile saline. Ensure that the plunger handle is completely extended to the back edge of the syringe barrel. Slide the plunger handle laterally to remove it from the process disposable. Be sure to retain the handle as it will be reattached after processing. Attach the green tip cap by screwing it onto the end of the processing disposable syringe. It is important to remove the plunger handle prior to attaching the green tip cap to ensure easy reattachment of the syringe handle after disposable processing. The process disposable may now be passed off the sterile field for processing on the SmartPrep device. Open the lid to the SmartPrep centrifuge and insert the AdiPrep adapters into each trunnion. Align the white dots on the adapters and SmartPrep rotor to ensure a proper fit. Place the processing disposable, green tip cap pointing down, into the sample holder in one of the adapters. Place the green balance weight into the sample holder in the remaining adapter. Close the centrifuge lid and start the centrifuge cycle. For SmartPrep 2 systems with serial numbers 101 to 5999, press the green Start button. For SmartPrep 2 systems with serial numbers 6000 and above, press the blue PRP button. The approximate centrifuge runtime is 4 minutes. While the minimum centrifuge time is 4 minutes, the AdiPrep disposable may be processed through the entire 14-minute centrifugation cycle with no negative impact on the adipose tissue sample. Once the display reads 10 on the SmartPrep 2 control panel, press the red Stop button to stop the centrifuge cycle. An audible alarm will sound once the cycle has been completed and it is safe to open the centrifuge lid and remove the process disposable. Remove the process disposable from the centrifuge. The lipoaspirate sample will have separated into 3 discrete fractions after processing. The infranatant fluid, present on the bottom layer closest to the green tip cap. The concentrated adipose tissue, present in the middle layer. The lipids, present on the top layer as a bright yellow fluid just above the floating lipid barrier disc. Reattach the syringe handle and remove the green tip cap. Expel the infranatant fluid into a disposable cup until the concentrated adipose tissue layer is reached. Be careful not to expel the concentrated adipose tissue out of the syringe. Within the sterile field, attach a female-to-female connector to a sterile treatment syringe. While maintaining the treatment syringe's position in the sterile field, attach the AdiPrep process disposable via the female-to-female connector. From the sterile field, draw the concentrated adipose tissue into the treatment syringe. Remove the treatment syringe from the female-to-female connector. leaving it behind with the processing disposable outside the sterile field. The concentrated adipose tissue is now ready for clinical use.